Hello, everybody. My name is Simon Warchol. I am a PhD candidate at Harvard. I'm primarily advised by Hans-Peter Pfister in the Visual Computing Group at the School of Engineering at Harvard. And then I'm also co-advised by Peter Sorger in the Lab of Systems Pharmacology in the SysBio department at HMS. And today I'm going to present on some work that I did along with my collaborators called SEAL, which is Spatially Resolved Embedding Analysis with Linked Imaging Data. This is, I guess, a longer version of a talk that I gave at ISMB earlier this summer, and then this will also be presented at IEEE VUES next month. So hopefully this is not the second time you're hearing it, but at least if this is the second time you're hearing it, you won't have to hear it a third time at VUES and can prioritize your time more strategically. So yeah, I am a computer scientist, but I kind of work directly with primarily pathologists and cell biologists who study cancer. And primarily they do this by investigating whole slide images, which are imaged through cyclic immunofluorescence. And so they're kind of the data that they look at on a daily basis are these huge gigapixel images that are hundreds of gigabytes in size with 30 or more channels, kind of each corresponding to the expression of a different biomarker. And then kind of from within these images, we can run segmentation algorithms to identify the individual cells that compose them. And so kind of from these single cells then, this allows us to kind of run this conventional single cell analysis pipeline. So what a lot of my collaborators do essentially, and I think what is pretty par for the course, I guess, in this space is you have an individual cell in this huge image, and you kind of look at, say, the mean expression of all the different biomarkers or the different channels in your image, and kind of create what they call like a quantification table that allows them to easily kind of summarize each individual cell in the image. And then from that, kind of following more standard single cell analysis analysis pipelines, then the dimensionality reduction becomes this huge tool where you can take these millions of cells and then quickly identify interesting subpopulations that exist within them. Typically this means looking at cell types or cells in different states. And so these 2D embedding then is generally used to visualize the results of this dimensionality reduction. And this is, as I said, really important because it reveals, some clusters that would be nearly impossible to identify just looking at the image. And, you can, again, see patterns. You can also notice outliers. And so, sometimes people use this as well for like artifact detection or something like that. You notice a huge outlier and it's actually because somebody's hair fell on the slide when it was imaged. And so you can, omit that section from the analysis or something like that. And so, yeah, clustering, running some sort of clustering algorithm is a standard way of kind of making sense of these embeddings. And so in this context, we have some clusters that were run on a single cell embedding, and somebody's annotated them to the such that we can see that individual clusters correspond to specific cell types in the overall image. And so, yeah, these embeddings are really, really useful and important, but I think often it becomes hard then to kind of link them back to the original spatial image, which, also contains some really, really vital information. And so, it tells you, where the cells represented in this embedding are, what they look like, and, what markers they express, which, and the expression of these markers is kind of more nuanced than simply a kind of mean of the expression value, which is often used in the single cell embedded analysis pipeline. And so the goal of this project SEAL is to basically try to bridge these bridge the gap between these two things. So, make it so that we have some of this imaging context and spatial context, while also, providing the benefits that the dimensionality reduction provides. And so more broadly, the data I'm looking at, which I've been talking about here, is kind of any multi-channel image where the individual entities within it have been segmented. And then we have some sort of corresponding embedding of that image, or of those entities rather. And so, of course, as I've been talking about here, this means any sort of multiplex imaging with corresponding single cell embedding. Interestingly, this is not these needs are not specific to this kind of single cell analysis task. And what we also found is this very closely resembles some of the ways that astrophysicists look at this multispectral astronomical imaging data. So they use kind of similar, looking at the different expressions of elements or wavelengths of light that are expressed to identify, stars or galaxies and whatnot. And so, and in fact, we actually kind of, in addition to collaborating then with some um, pathologists and cell biologists, we also then kind of established a collaboration with the Harvard Smithsonian Center for Astrophysics. So again, worked with some astrophysicists as well to kind of try to generalize some of the analysis workflows and visualization needs that are shared kind of across these domains. And so, yes, in direct collaboration with these kind of two main groups of users, we kind of went through this design study methodology to identify some requirements that these experts need when they're, kind of going about their job. And specifically in kind of looking at, two-dimensional embeddings and kind of bringing them back to the underlying spatial image. And so, first of all, they need to kind of situate the data, kind of both in the embedding and the image. So, where does a cell, where does a planet exist in the embedding, and then where does that exist in the image, and how can we kind of link between these when we're looking at, clusters or regions of interest in either. They want to be able to then select kind of sets of data based on different criteria. Again, this could be a clustering, this could be a combination of clusters, this could be a lasso selection or kind of any arbitrary list of entity IDs. And then they need to be able to characterize these sets of items that they've selected in terms of their distinguishing features and attributes. So we see a scatter plot, UMAP scatter plot, and we see there's one cluster that's really separated from everything else. But what's important then is to understand why it's so separated from everything else. Presumably there's something in the underlying feature data that explains why it's why it's the algorithm has kind of thrown it all the way over there. And so this is kind of a key need from these domain experts. And then finally, they need to be able to compare sets of items. So understand, how one cluster is maybe different from another cluster, or one, region of interest is different from another region of interest. And so, as I think a lot of the people on this call probably are aware, the existing analysis landscape, kind of contains both viewers that people use to kind of look at the images. So in the cell imaging space or the multiplex imaging space, a lot of my collaborators use something like Omero or QPath or ImageJ to look at the images themselves, or use kind of more dedicated viewers. Our group's been working on a system called Minerva, which is kind of a storytelling system for bringing people through the images. But then when they're actually doing kind of computational analysis, then that exists kind of all in this Jupyter scientific Python kind of environment. So they're using, I think, ScanPy, SquidPy. A lot of those systems are very, very popular for doing single cell analysis and I don't know, clustering and faceting the data. And I think interestingly, this like analysis kind of goes. I guess this honestly should be like a cycle, and that people go between the dedicated systems for looking at the images to kind of doing some more computational analysis. I think that's like interesting and probably worth a kind of greater discussion as to why like these things have been kind of siloed out in the way they are. But I think that is something that a lot of the folks here have probably been working on addressing. I know that Niels's group has specifically kind of worked on systems that kind of are in the middle of this. So we have then we have Vitesse, we have Napari, we have systems that kind of try to bridge this gap to some extent, and I think have been relatively successful in doing so. And so we're kind of situating this work then kind of in the middle there, and specifically we're kind of building on some of the views that Vitesse offers to kind of as the backbone of the analysis. And so kind of what this looks like then is this system that we've developed kind of as deployed both kind of as a standalone web app then, and then also using any widget kind of by Trevor Mans, who is I guess formerly in the HiDi group. We also then can easily kind of deploy it as well as a Jupyter widget. And so instead of, needing people to kind of take their data and, bring it over to some totally disparate environment. Instead, they can pass in a pandas data frame as a means of analysis and kind of run clustering in an iterative manner as opposed to, having to have these really, I don't know, stark handoffs, I guess, between systems. And so I think this is, definitely the way that a lot of the research and kind of tool building has gone, is to try to bridge this gap. But I think there's still kind of a ways that we can go in terms of really making it so it really fits the needs of kind of, computational biologists. Um, and I think one thing that we do that's kind of specific then to our approach that I'll get into in a second is we really look at these kind of as I mentioned earlier, we view these clusters and selections in the embedding or in the spatial image as sets. And so this allows us to employ, set visualization techniques and mathematical operations. And so a set could be, as I said earlier, the result of a clustering algorithm, multiple different clustering algorithms. Perhaps you're doing some, hyperparameter tuning. You're doing k-means, k equals 5 with k equals 10 or something like that. This could also be, a region of interest, either that could be, again, a region within an embedding space that you think is interesting and perhaps wasn't totally isolated by the clustering algorithm, but you still want to look at. Or this could be, some sort of biological feature in your original image. Any selection of entities here is a set. And then again, this allows us to kind of look at these set comparison algorithms kind of as a means of identifying, what makes one set different from another. What are the shared entities between these sets? What are things that are totally omitted by the sets? These kind of standard operations. And so, yeah, with that, I guess I will get into the system that we've developed. And kind of throughout the next, as I kind of show some of the capabilities of the system, we're going to be looking at a section of colorectal cancer tissue, which was imaged by one of my lab mates, Jerry Lin, at the Lab of Systems Pharmacology. And I think this specimen, I think, is particularly interesting to look at because it's, been really, really well studied by a lot of my collaborators. And I think more specifically, Greg Baker, I guess, is now developed a kind of a system that built on Napari, um, that uh, that that looked called Silenter, which looked at how we can do kind of quality control and on multiplex images. And what he basically published, and as part of the publication in Nature Methods, he had just a lot of really, really well annotated kind of labels on this UMAP embedding that he generated. And so kind of using that as some sort of, ground truth in a way, it has been really, really helpful and allows us kind of to, see basically if some of the methods and visualization techniques that we're developing kind of are in fact capturing kind of the underlying biological properties of the data. And so this system then kind of has three key views. I think the first and kind of least exciting or surprising is just a standard kind of image view in the top right here, which just is yeah, basically, Vitesse, right? Like or Viv, I guess, under that. But to show allows the user to kind of look at, multiple channels simultaneously, zoom and pan around and whatnot, and kind of reads from a variety of file formats. Kind of more interestingly, then, this kind of second view in the top left here is something we're calling a hybrid embedding view, which is essentially essentially kind of we're trying to improve just simply using a scatter plot as a means of visualizing some sort of dimensionality reduction technique. And so this embedding then corresponds kind of directly to this, in this case, it's a UMAP that was run on this single cell data. But instead of kind of showing individual points in the UMAP, instead what we do is we kind of cut out each of the cells in the original image. This is done using a tool called Cell Cutter that was also developed by a colleague in the Lab of Systems Pharmacology that kind of is also very useful for if you're doing any sort of kind of trying to create training data for any sort of machine learning algorithms. But basically, we cut out all the cells, and then we try to place the cells at where they exist in the embedding, as opposed to in the original image. And I think one issue we run into there is whereas, in a tissue, we wouldn't expect there to be 100 cells directly on top of each other in one 2D slice. And in an embedding, we have no such, guarantee. There's some hyperparameters that people use, MinDist or something like that in UMAP or TISNE that people use. But again, you don't really have a great way of guaranteeing that there aren't, going to be areas of really, really high density that, so what we do then is we kind of take this multi-resolution non-occlusive placement approach where we create basically a new kind of hybrid embedding image that is essentially the same kind of size as the original image. And then at each kind of level of resolution, so as we move kind of down the image pyramid, we try to place basically as many cells as we can until they begin to occlude each other. And then once there is occlusion, then we kind of choose not to place that cell, and instead, we'll then place it at the lower and lower level. And so, at all the way zoomed out, then we're only showing kind of a subsection or subset rather of all the cells in the embedding. But as a user kind of zooms in, then they can see all the underlying cells entities that make up this image. And so, what that looks like then is, again, so as you zoom further and further in, more and more cells are then, rendered, and you can see how they're kind of packed in there to avoid any sort of occlusion. And, if you zoom then all the way in, basically all the cells then should be visible. I think one limitation to this approach, I suppose, is that it can be hard from this original embedding, and I think this is a big limitation of scatter plots as well, is it can be hard to identify kind of regions of high density. And so one thing we also added is then the ability to kind of have a contour plot that shows density. And then finally, you can also choose to plot kind of each of the individual dots, kind of like a scatter plot, in case there's any sort of confusion. But I think that we would assert that showing the individual cells is much more useful, in that it shows their marker expression, and so it shows their morphology. Whereas these little dots in the scatter plot don't, they're basically just showing their position as well as some sort of class label. So yeah, both of these spaces then can be, analyzed, like any sort of multiplex image. So you can, toggle multiple channels and, zoom in, pan around. You can, select regions of interest kind of with a lasso tool. So again, pretty standard. Oh, see that again. Yeah, so selecting kind of regions of interest with the lasso tool, and then you can, in doing so, it should. Okay, well, I guess, yes, sorry. So when you can select region of interest, the lasso tool, I think then the question becomes like how we're going to highlight basically where members of that set exist in the image. And so I think there's a lot of different, standard kind of visualization approaches to doing so. I think outlining them with, using the segmentation mask is probably the most standard approach, in that it's, at least non-occlusive to the imaging data within the segmented entity. I think one issue we see, especially as we're looking at kind of depending on the contrast limits that a user has set, the colors that they have assigned to the individual channels, and just the number of channels they're visualizing, is it can be quite hard to see sometimes these outlines. And so, you could totally miss, a pattern just because, it's in a kind of relatively bright area. I think these cells in the bottom left here with the green background are kind of quite hard to see. And so we also offer kind of some alternative ways of visualizing selections, namely, I guess, through spotlighting. So essentially, instead of kind of drawing any sort of segmentation mask or on the image, instead what we do is we basically choose to like darken the anything kind of outside of that selection. And I think a user can choose whether that means to kind of and you can change the opacity kind of the spotlighting. So basically you can choose to kind of darken the regions that correspond to cells that are outside of the selection, or you can also choose to darken the area of cells and kind of any unsegmented region, so any kind of background or other things there. And then you can combine these kind of as a way of better identifying kind of where your sets exist kind of in both of these two image views. And so, yeah, as you said earlier, you can lasso kind of a region of interest, which I think we've done down here. And it looks like the the cells that correspond to it are in this kind of far right region of the image, but that's kind of hard to see, I think, from the original view. I mean, as you zoom in, it becomes more and more evident, but then we have this issue where, now we're kind of occluding what the actual cells are in that region. And so you can then choose to, instead spotlight that selection. So, through the kind of view panel down here, you can, choose to kind of darken. And here we've kind of picked a kind of, I don't know, 50% opacity is kind of the limit for the selection, but that you can kind of darken the regions around it. So this makes it, a lot more evident, I believe. And finally, you can, as mentioned earlier, can choose to kind of toggle the background as well. So you can really then, choose to kind of exemplify or emphasize where these cells exist kind of in the tissue or whatever spatial entity you're looking at. And finally, as mentioned, you can always turn off these outlines. So again, you now just see the kind of cells with no, occlusion at all in the markers that they express. You can also look at their morphology kind of much easier. And as mentioned earlier, you can, toggle kind of how dark you want this kind of spotlighting to be. Okay, finally then to move on to this kind of third view at the bottom of the page here. And so this we call the set view, and this is where we're visualizing all the different sets, again, which could be regions of interest, lasso selections, clusters that a user has defined or imported into the data. So this is what, when you're doing this in Jupyter, you specify like cluster columns in your data frame, and then it will kind of visualize those as different sets in this view. And so to kind of walk through what we're actually visualizing here. So each of the individual, so each of these columns then, so this is an individual column I'm seeing here, corresponds to an individual set. And then each of the rows right now is visualizing the feature importance of the different features to that set. And so in this case, you can see that this set is mostly defined by pan-CK as well as ecadherin. If I mispronounced that, I'm sorry. I'm a computer scientist, not a biologist. And what you can do then is at the top of the page as well, it shows kind of where those cells exist, either in the embedding, or they can kind of flip this to show where they exist in the image itself. And so what then do we mean by feature importance, or how do we define it? And I think that's something that's quite tricky with a lot of these dimensionality reduction algorithms, is that they can kind of be black boxes to a certain extent. I mean, of course, there's something like PCA, which is, I think, very interpretable, but I think just doesn't do the same preservation of both local and global neighborhood structures that something like stochastic algorithm like t-SNE or UMAP does, and so I think is a lot less common. And so what we propose as a means of calculating feature importance then is by doing so through kind of a surrogate model approach. So what we're essentially doing is building a model that is trying to predict or trying to emulate the underlying dimensionality reduction algorithm to the best of its ability. And so let's say we have some, single cell embedding as well as the kind of corresponding feature data. We train an XGBoost regressor, which is a tree-based regression algorithm, kind of based on these features. And so what that looks like then is, so we have, each of these, as I showed earlier, each cell which corresponds to, which is represented by the mean expression of a different, channel, different biomarker. And so we basically train this regressor to predict basically where in the embedding each individual cell will exist. And so, in doing so, we can kind of create a rough estimation of the original embedding. And I think it's also, I guess, worth noting that I think there are some other approaches to doing this that I think also are useful. I guess more specifically, there are kind of different dimensionality reduction algorithms that would offer kind of more, they would offer kind of native feature importance. I think parametric UMAP has a place here, but I think that we found that often the analysis that people are conducting is, they already have some sort of, they have a dimensionality reduction algorithm that they know and they trust, an embedding that they're interested in investigating, or perhaps it's the embedding that somebody put in the figure of their paper, and another scientist then is trying to do some follow-up work and understand kind of why the global landscape of cells in this tissue looks the way it does. And then a real benefit to using this kind of XGBoost regressor here is, again, as a tree-based algorithm, this allows us then to easily calculate SHAP values then on kind of each individual point or a selection of points in the image. So if we have, say, this ROI, this cluster in this embedding, then we can easily compute kind of the mean SHAP values for the cells in this cluster. And then, in this kind of case here, we see these three biomarkers were kind of the features with the highest importance. And if we, plot them kind of just as scatter plots or whatever, we can see that, in fact, yes, the, pan-CK in the far left has, you can see it's much more expressed in this region than anywhere else. But I think one limitation we see, or not a limitation, but one thing we don't have a great way of knowing strictly from looking at these mean feature values is if it's kind of a negative or positive feature importance. So as you can see in the far right, yeah, that's pan-CK. You can see that in fact is not expressed at all in the region of interest. And so that's something that we also kind of encode, which I'll show in a second. Or actually, yes, so you can see right here in this kind of selection. So in addition to having this color scheme, which encodes the feature importance with the Viridis color map, we also draw these little lollipop charts on each of the columns. And so the lollipop charts then encode the relative expression of a given feature within this region of interest. And so, if we have lollipops going to the right, that means that feature is expressed more on average relative to the overall data set, or if it goes to the left, it is less than average. And then you can also kind of choose to switch these encodings such that instead we use kind of a diverging color scale to encode positive or negative relative occurrence, and then use the lollipop strictly kind of, which again are only kind of, yes, it's an important feature, no, it's not an important feature, encode the feature importance. And then finally, I think one thing that, doing a lot of this work, which, basically takes the means of a lot of values, could make hard to identify is that if, let's say, you have a set that has, or a selection that in fact kind of contains two interesting subsets that in fact have like different different occurrence of a different feature. And so finally, you can also then switch this view to show the distribution of these individual values within this set. So if you have, two humps in there, you'll see that, oh, this set actually might have two interesting subsets that are worth kind of further investigation. Cool. So then kind of from this original set view, kind of the basic functionality, you can just hover over them to show where the cells that correspond to them exist in the image in both of the two images. Again, you can also turn on the spotlighting or the other kind of view modes that highlight these entities more. You can, we also kind of offer, we do some annotation on the embedding or the image view. So for any kind of for any set that has a density above a certain threshold, we draw a concave hull around it, and you can annotate that on the image on either of the two images, and then also you can optionally choose to show the kind of most important features on top of that below the annotation as well. So let's say you're interested in this particular, set. You can click on this set and again show where the images, or where the, in this case, cells exist in the image that correspond to that set. Another thing you can do is, let's say you're interested in a set, not just because you want to know kind of where those, in this case, cells exist in the image, but perhaps you think that you're interested in what cells might be around those cells, which is something that some prior research of mine has also looked into. Is a really important kind of analysis task for single cell biologists. And so you can also choose to kind of do to create a new set that corresponds to the nearest neighbors of a given set. Either in this nearest neighbors calculation can either be done in the image space, so what are the cells around each of the cells in a set, or in the embedding space if you're selecting from the image space. So finally, as mentioned earlier, once you have these sets, it's interesting to compare them, to understand what makes them unique. And so let's say I have these two sets here that I want to compare. You can then, by selecting two sets and clicking this compare button, then we'll create new sets that correspond to each of the set operations that have some number of entities. So in this case, when you have two clusters that have no overlap, so no shared membership. Basically, I mean, you're only going to see the union and the complement of those sets as, everything is either in one, in the other, or not in either. But perhaps we have some region of interest where, we're interested in kind of some area that has some overlap. Then you can similarly kind of choose to compare these two. And in that case, then we'll also visualize what entities are only in the first set and not in the second set, what's the symmetric difference of the two, what's in the intersection of the two. And I think this offers a way that a analysis task I saw a lot is basically people would have some sort of cluster in an embedding space and want to do kind of lasso selections kind of for like I don't know, the whole set, these two parts that overlap, what's in the middle of it, and kind of try to make sense of, what the axes of information within an embedding are. And so I think this provides some sort of means of doing that. So this is all in a lot of this kind of analysis or this evaluation stuff is much more covered in the preprint, which I'll show a link to at the end. But in the corresponding paper, then we kind of offered two or a few different case studies where we worked with domain experts and had them use the system and analyze their data with it. I think the first one, which we showed a lot here, but it is covered in more detail in the paper, is basically looking at this colorectal cancer tissue that again has been really well annotated, kind of as part of a previous publication, and identifying kind of if the feature importance values did in fact correspond to what was defining these individual cells. And we found that yes, it was very, very easy to identify interesting clusters or regions kind of using the approach. In the second evaluation that we offer, we looked at some melanoma tissue, and this had been imaged with both H&E as well as immunofluorescence imaging. And the scientist here was training a variational autoencoder to try to create a shared embedding space of both this H&E data as well as this IF data as a way of kind of understanding the correspondence between the two of them. I think the pitch was kind of like some sort of multimodal embedding like clip or something like that, except for just IF and H&E. And in that we're able to very easily see kind of different regions in the embedding that correspond to different states, necrotic cells, non-necrotic cells, and other biologically important features, which again is further covered in the paper. And then finally, we met with an astrophysicist to look at sky survey data, which is a big, huge data set that images the night sky at a lot of different wavelengths. And there we were and that, I guess, as well is covered in the paper, but there we were basically able to identify galaxies, like clusters that corresponded to different stars of different ages and galaxies with different morphologies based on kind of investigating this hybrid embedding space. Cool. So all of the preprint as well as some of these demos are all available on the website. It's sealviz.com. I'll throw that up in a second, but kind of from the website, you can look at there's a small data set of a tumor microarray that you can analyze. We also have this Sloan, this sky survey data where you can look at some of this galaxy information, the stars within it. And finally, we also have an example that embeds the notebook in Colab, so you can install the package and then analyze the same. I think this should work for both of the two exemplars within a computational notebook. To, I guess, quickly cover the architecture, a lot of this is built on some of the really great work that's been done by Niels and his students and the rest of the groups. I'm really thankful for that. Specifically kind of this Vitess Viv and any widget. And so again, this runs on a kind of Python backend that also runs on the server, but with Vitess as kind of the primary or first building block of the front end, and then again deployed kind of just the same code base very easily kind of can be deployed to both places with basically one build command, which is really I think really nice to not have to think of these as like totally separate development workflows. Yeah, so to cover some of the kind of things we're thinking about next. So I think right now the data I look at, or the data my collaborators look at, kind of has this assumption that there's a mapping between these features and image channels, right? So basically all of the features in the single cell table correspond to, this image channel. But I think that that's totally not the case in in other modalities, or if you had more analysis where you were quantifying, say, morphological features. You can we have some we look at the roundness of cells, or size even. And so I think that integrating those features in a better way, I think, would be really useful. I mean, at a base level, that's just like putting maybe some heat maps or something like that on top of the image. But I think there's probably more interesting ways that we could really visualize these kind of derived features or non-spatial features within the context of the imaging data, kind of both from the embedding standpoint as well as from the underlying image standpoint. I think one thing that also is kind of a key limitation here is we're basically viewing all the sets on this kind of flat hierarchy. But, in fact, I guess, what you'll find is, when you, have, let's say you have two different clustering algorithms that you run, and, one of them, creates this like blue cluster in the bottom left of this scatter plot, and then one of them, separates it into some number of other clusters. Instead of viewing them kind of all in the same flat hierarchy, it's much more useful to like think of to actually, formalize the relationship, formalize the hierarchies that exist between sets here. And so I think that's something that I think both from I think that's something that is pretty easy, or low-hanging fruit strictly from a kind of user interface and interaction standpoint. But I think that from an analysis standpoint is something that I think still needs a lot of work to really factor that into what you're doing. And also kind of formalizing the relationships that might be like, oh, these, if we really think that a dimensionality reduction, like if we think that two clusters being adjacent in the dimensionality reduction does correspond to something that's scientifically meaningful, how can we formalize that these two things, that these B cells and T cell clusters should in fact be near each other, because like those cells have some sort of key interaction that goes on between them. And so formalizing that as well is really important. And then I think the other thing that I'm really interested in is kind of how we can identify and visualize basically trajectories, either through this embedding space or through the image space. I mean, one thing we'll see when we look at, like when we look at kind of how immune cells express relative to a tumor, we kind of think of this, there's like basically a gradient, that goes from, far away from the tumor to right adjacent to the tumor, where cells are kind of moving in and out of the region kind of as they become exhausted or activated and whatnot. And so I think that providing kind of more ways for people to understand these trajectories of these spaces is really important as well. And so, that also can be something that's interesting to look at kind of within a cluster itself. I think that people often notice that, throughout some sort of axis in a cluster, there might be, towards the top of this are subsets that express a certain marker more into the bottom. Again, maybe it's exhausted versus activated immune cells of some sort. And so kind of having a way that you can really kind of sketch these trajectories through these spaces and visualizing it, I think would be really important. Both within the embedding as I show here, but also in the image itself, where these trajectories correspond to the actual movement of cells. And then, of course, as I'm sure every future work section that looks at 2D imaging contains, yes, looking beyond single 2D data sets, be it some other work we've done where we look at cohorts, but I think more interestingly, of course, looking at the 3D stuff, which I'm sure Niels's group has presented a lot on kind of in this meeting in the past. So with that, yeah, I'd like to thank kind of all of my collaborators who helped in this project, as well as the kind of funding sources. And yeah, this sealviz.com, hopefully the AWS outage yesterday did not impact it negatively, and it's still up. But I'm really happy to answer any questions, hear from anybody, collaborate. Yeah, just thanks to everybody for being here.
Well, thank you so much, Simon. What a great presentation.